Hello again, everyone. This is Adam Paulson welcoming you back to our final Lab Meetings Live presentation today, brought to you by Bitesize Bio. Our final keynote presentation today is titled Precise High-Efficiency Editing of Stem Cells to Probe Human Biology and Model Disease, and is being presented by Professor Bill Skarnes from the Jackson Laboratory for Genomic Medicine. Bill received his BSc and MSc in Microbiology and Immunology from McGill University in Montreal, Canada. In 1992, he was awarded his PhD in Molecular and Medical Genetics from the University of Toronto, where he pioneered gene trapping technology in mouse embryonic stem cells. In 1997, Bill took up an appointment as an assistant professor at the University of California at Berkeley. From 2003 to 2016, Bill led the mouse developmental genetics and embryonic stem cell mutagenesis teams at the Wellcome Trust Sanger Institute in the United Kingdom, focusing on the generation of a genome-wide collection of conditional knockouts in mouse. He has over 80 peer-reviewed publications and is now based at the Jackson Laboratory for Genomic Medicine in Farmington, Connecticut, as the Director of Cellular Engineering. Bill's laboratory is currently exploiting new genome editing technology to study gene function and model disease in human stem cells. We will have a question and answer session after the presentation, so please type any questions that you have into the questions box which appears on the bottom of your screen, and I'll put them to Bill at the end. So now over to you, Bill, for the presentation.
Thank you for the introduction. Today I'll be talking about two technologies, both of which have been awarded Nobel Prizes, which when combined gives us a very powerful platform to understand basic cell biology, early human development in a model cell system. These two technologies include reprogramming, which is used to create stem cells from somatic cells, and the so-called induced pluripotent stem cell, and CRISPR technology, which is we can use for editing the genome of these induced pluripotent stem cells. Now, one of the big advantages of induced pluripotent stem cells is that they can be differentiated into virtually any cell type of the human body, and they can be propagated in culture indefinitely and will maintain a normal diploid karyotype. So these are normal cells, unlike those cells that have been used in the past to address human cell biology and biochemistry, which includes HeLa cells or HEC293 cells. These cells, in contrast, are highly abnormal cells. They don't have a normal genome, and so it might be a good time to revisit a lot of the cell biology and biochemistry that we have learned over the past 40 or 50 years, but in a normal cell like a human induced pluripotent stem cell. So the second technology, editing, makes possible the modification of these cells at the genetic level, so we can introduce mutations into these cells. And the incredible property of CRISPR technology is that one can make biallelic mutations, so one can modify both copies of the gene simultaneously. With conventional gene editing technologies that were used in mouse embryonic stem cells, this was not possible. It was only possible to modify one of the two copies of the cell in a single round of editing. So by combining iPSC with gene editing, one can imagine using these cells to phenotype these cells in a variety of different ways to address different areas of human biology. So we have a vast amount of human biology that can be accessed using human iPSC cell technology and genome editing. And so what I want to focus on today is a project that we are currently running, which is looking at the effect of single nucleotide variants on neurodegenerative disease. This is the so-called INDIE project. So this is an NIH-funded project, which is led by Mark Cookson and Michael Ward at the NIH. And the idea is to introduce into a healthy iPS cell line, a normal iPS cell line, mutations that have been associated with neurodegenerative disease, specifically Alzheimer's disease and related dementia. So we can use gene editing to introduce mutations or single nucleotide variant mutations that have been found to be associated with these diseases. And then we can also make a number of companion lines that help in terms of the analysis of what the gene mutations might be doing in differentiated iPSC neurons, glia, and astrocytes. So this is a relatively large-scale program, and so it demands high-efficiency editing technology in order to carry out these numbers of projects. And the guiding principles that we adhere to for these editing projects are as follows. So, and this is generally important for any editing project in any cell line, is that you want to make sure that the cells that you start with are relatively homogeneous. And so the first step to any editing project should be subcloning the cell line, and this will reduce genetic heterogeneity in the edited clones. So if you imagine that the cell line contained a variety of different mutations in the background of these cells, and that they're not essentially clonal cell lines, that you will separate out these mutations in individual clones after editing, during the editing process. So we feel it's really important to subclone the cell line that you're using for genome editing. A second principle is that we really need to characterize the iPS cell line that we want to use very carefully. We want to make sure that it has an intact genome, so we will use karyotyping, high-density SNP arrays, and even whole genome sequencing to characterize those cells. We're also checking for mutations in T53, which are often mutated in culture of iPS cells, to make sure that they have normal function of this important genetic pathway. And we want to look at their differentiation potential. So for neurological diseases, we want to make sure that these can differentiate into a variety of different neural subtypes, which include neurons, astrocytes, and glia. In terms of the edit itself, we want to make sure that we are only making the mutation that's found in nature in the human population with no additional changes. So we know that one can introduce silent mutations which may improve editing efficiencies, but the risk is that these mutations could affect the property of the gene, transcription of the gene. So some missense mutations, for example, are known to create splicing problems. So we want to make sure that we make only the disease mutation, which in most cases involves just a single nucleotide change. Once we've made those changes, we want to assess the genome of post-edited clones, again, to make sure that these are normal after this editing process. And so we will look for on-target effects. We'll karyotype the cells again. We'll look at their genome by high-density SNP arrays. I'll explain what on-target effects are in a moment. And finally, as a control for these experiments, once you identified a cell with a phenotype, you'd like to be able to prove that that phenotype is due to the mutation and not something else that's been acquired during the process of editing or culture. So the last step of this process would be to revert the mutation using CRISPR editing tools to control for any off-target or cell-acquired mutations. So just to drive that point home, we want to be able to fluently engineer both mutants and revert alleles. And so this could be visualized by this slide, which shows that initially we'll take wild-type cells and introduce the mutation using a mutant oligo and a Cas9 RNP. Once we've made that mutation, we want to revert it, so we will then introduce a Cas9 RNP that recognizes the disease allele and a wild-type oligo to revert that mutation back to wild-type sequence. And so in this process, we need to avoid introducing pan mutations. So we will select guides that do not introduce a pan mutation in the locus. And finally, if you're interested in any of these cell lines that we have been developing, you can go to this website at the Jackson Laboratory, and you will see sets of genes, sets of clones, which include the disease model, which in this case would be SNV over wild type, the revertant of that very cell line, which is the revertant over wild type. And as an additional cell line, we also offer, in this case, the homozygous SNV over SNV cell line. So we'll be adding to this collection over the next year or so to generate a very large collection of over 150 different disease variants that are associated with Alzheimer's and related dementias. So the choice of the reference iPS cell line project was critical. And so we looked at 8 different iPS cell lines for their properties and looked at their genomes, their differentiation properties. And we settled on one cell line, which is called COLF 2.1J, which is a cell line that I've been working with for many years now. We know that COLF 2 cells are very stable in culture. They can be easily edited. They have a broad differentiation potential. So these cell lines are male. They come from an individual of Northern European descent. This is actually an edited clonal subline that we start with at low passage. And these are factor-free, so they've been reprogrammed with factors that are now eliminated from the genome using Sendai virus technology. And most importantly, these cells also have open access to any genomic information that we collect on these cells. So COLF2 originally come from iPSC, which was a large-scale iPS derivation program at the Wellcome-Trust-Sanger Institute. And we've been working with these cells for some time, and then we're notified that the cells actually contain a pathogenic variant, 2 pathogenic variants, but one of particular interest and concern, which was a pathogenic variant in the ARID2 gene. So this is a deletion of 19 bases, which introduces frame shift mutation in 1 copy of ARID2. And in humans, we know that mutation of ARID2 in a heterozygous state causes intellectual disability. This is the so-called Coffin-Cyrus syndrome. So we wanted to correct this mutation before we started this project, because obviously we don't want mutations that are in the background of the cell to confound the phenotyping of cells that carry mutations that cause neurodegeneration. So we use CRISPR editing then to correct these cells. And I'll just point to a second pathogenic mutation that we did not correct, which we didn't feel was important in neurological diseases, which was a collagen 3A1 mutation. And so that's still in the background of the cells. But I think this is fairly typical of any iPS cell lines that you're going to identify or find spontaneous mutations which have arisen either during the reprogramming, during culture, or even in the starting cells that you start reprogramming. So this is a display, a tool that we use to identify guide RNAs. What I'm showing you here is the region of ARID2 that we corrected. So you can see that there are tracks that show the variants. So the COLF2C11 and COLF2C112 are the starting parental cell line, and you can see that there's a deletion of this 19 base pair sequence in the middle of this ARID2 exon, which we corrected. So the variants that you see on the COLF 2.1 tracks are the variants that are present after sequencing the corrected cell line. So that mutation is no longer present, but you can also see that we have maintained heterozygosity. So we know that there are 2 copies of the corrected gene in these cells. So finally, after assessing the genome of COLF 2.1 corrected cell lines, the so-called COLF 2.1J cells, we wanted to make sure that they still maintain their differentiation potential, specifically for neuronal cell types. And so we passed these out to a number of labs to compare these cells with the cell lines that they're generally working with in the lab to make sure that they can differentiate well into a variety of different neural subtypes. And in all cases, these labs have shown that the COLF 2.1 cell lines are very good at differentiating into different neural subtypes in culture. Okay, so now to move on to the actual editing of these cells, I think everybody is familiar with the fact that Cas9 or Cas12a, these CRISPRs are RNA programmable programmable nucleases, and so these nucleases will recognize a specific site of the genome and make a double-stranded break. And then that double-stranded break can be repaired by one or two pathways. One is by non-homologous end joining, or NHEJ. This is an error-prone process, which gives you indels at the site of cleavage on one or both alleles. And that's been useful for certain kinds of projects where you want to, say, mutate the gene. But we want to be more precise. And so what we can do is we can add a donor template, which will then get incorporated at that break point and introduce, in this case, a single nucleotide variant at that break point. And this is through a process known as small-directed repair. When we started these experiments, we found that about 10% of unselected cells after nucleofection carried the single nucleotide variant on one or both copies of the gene. So this was relatively inefficient for a large-scale project. So we spent some time testing different conditions to try to optimize the editing of these iPS cells by homology-directed repair, and we turned to a classic BFP-GFP assay where we are monitoring homology-directed repair through the conversion of BFP to GFP through a single nucleotide change. So if you change C to a T and a histidine of BFP, you end up with a green fluorescent protein, GFP. And then this gives us a very easy assay to measure editing efficiency, both of HDR and NHEJ. And the FACS sort plot that you see on the right there shows you about the efficiency that we were getting when we started this project. About 10 or 20% HDR was possible at that time. So we tested a variety of different conditions and treatments, and we found specifically 3 treatments that really improved HDR efficiency. And these 3 conditions are additive, so each on their own improve, but you combine them and then we'll improve them in an additive fashion. So the 3 things that we found that have really improved HDR efficiency was the use of a small molecule enhancer of HDR called HDRE. We found cold shock also improved efficiency on its own, but in combination with HDRE improved it even more. And then finally using N-modified single-stranded oligodonors, which are more stable. In addition to cold shock and HDR, we could really boost the efficiency of HDR to, in this case, about 68% of cells. This work is published, and you can read more about how we do these treatments and the methods that we use for editing in this 2019 methods paper. Since then, we've made a few other small modifications to this protocol, which I've shown you here on the right. So what we're doing here is we're using a MAXA nucleofection system. We're using high-fidelity Cas9 recombinant Cas9 protein. We add in the guide RNA, which is synthesized and provided to us by Synthego, and we use these N-blocked oligonucleotides from IDT, in addition to the treatment of the cells post-nucleofection with the HDR enhancer and cold shock. And under these conditions, we could actually see that HDR efficiencies now exceeded about 90% of cells. And what's also really interesting is that we didn't require pre-assembly of the Cas9 RNP before nucleofection. So you can just add it all together into the P3 buffer, nucleofect the cells, let them recover, and then we can do the FACS sorting or the single cell cloning at that point. So we're not doing anything other than delivering these reagents and culturing under these conditions. We aren't using any methods to select cells or enrich for the cells that are modified. These are just unselected cells that survive nucleofection. So to talk a little bit about how we design these projects, I've just shown you one example of the mutation in the APP gene, the A637T mutation, which is a single nucleotide change, which changes alanine to threonine. So we can go to that site using this tool, WGE, and identify guides that overlap this site. And what you'll see here are there's just one guide that overlaps the site, which is shown here. And we then inspect the off-target score because we want to look for guides that have few sites that are related in the genome. So the off-target score tells us that there's one site with no base differences, which is this target site. There are no sites with one mismatch, no sites with two mismatches. There are 5 sites with 3 mismatches and 4 sites in the genome with 87 mismatches. 87 sites in the genome with 4 mismatches. So this is a very good off-target score. And so we tend to use guides which have no matches with 1 or 2 mismatches in the genome. So this looks like a good off-target score. So this is now showing you the sequence of the gene and the guide. So you can see there the guide is highlighted with the PAM sequence on the right. And then we introduce the single base change that converts alanine to threonine, which is a G to A change. And we synthesize all of the nucleotide with that change. And for these projects, we've been using asymmetric target strand oligodonors, and you can see how those are designed here. So everything that you need for these experiments, you can buy commercially. And then we transfect the cells, we perform single cell cloning, and then we do Sanger sequencing. So this is actually a pretty impressive example of what we can produce with this editing, under these editing conditions. So the change that we're making is in the very first base of the guide. This is outside of the target seed region of the guide. And one would expect that it might be susceptible to cleavage after incorporation. But even with this position, with this overlap at the very end of the guide sequence, the very beginning of the guide sequence, we're still able to recover good numbers of heterozygous and homozygous clones for this single base change. So we've developed a high throughput editing workflow that allows us to carry out nucleofections in parallel. So we use a 16-well a max cuvette to transfect a small number of cells. We let those cells recover, and then we freeze those pools. And then we can thaw these pools at will to start single cell cloning in isolation of clonal cell lines that carry the mutation. So for single cell cloning, it's very rudimentary. Just plate out a small number of cells on a 10 centimeter dish, and then we wait for the colonies to appear, and then pick colonies, and we then distribute those colonies onto 2 dishes, onto 2 96-well plates. One that we freeze and archive, and the second that we lyse for PCR. So we can maintain these cells in culture for a short period of time, and then genotype those clones. And once we've identified the clones that have the mutation we want, we can thaw them from the matrix plate, expand them for further characterization. I should say that one of the things that might be important is to test guides in vitro before selecting the guides for the experiment. So we run a simple in vitro testing of our guides in cases where we have more than one guide that overlaps the mutation. So this shows you that the results of screening pairs of guides for particular mutations, and the ones that we've chosen are starred. And you can see that in some cases, for example, a UBQ gene there's a guide 7 that does not work very well. You see very little cleavage in vitro, whereas the second guide, guide 4, works quite well to cleave that fragment of DNA that contains the CRISPR site. So we think it's worthwhile to pre-screen guides and then to choose the guide that's relatively most active in this in vitro assay. So when we look at the data that we're getting from these experiments under these optimized conditions, it's quite remarkable that we are seeing in many cases, in about 2/3 of the experiments, a very large number of homozygous biallelic HDR events and very few wild type or heterozygous clones. So under these conditions, for the majority of guides, we see very high rates of biallelic HDR. But the outcome of the experiment is very guide dependent. So on the far left, you can see a guide that gives us a good mixture of heterozygous and homozygous clones, which is in our experiments a smaller proportion of the guides give us this pattern of editing. On the far right, this is a kind of exceptional case where we think that the guide, that the mutation that we've introduced is actually lethal to the cell. So we do not recover homozygous clones. So we get high efficiency editing, but we don't recover homozygous clones. So that's just, we suspect that mutation is actually causing cell lethality in iPS cells. So just to give you more data on these experiments, you can see the first set of experiments that we ran gave us a very high number of biallelic events, SNV over SNV, with very few, if any, heterozygous or wild type clones. So that's quite remarkable. But as we go down the list, you can see that efficiency does drop for some of these projects, and when it drops below 50%, we start to struggle to get homozygous clones. So as I said, these experiments are guide dependent, depends on their activity relative to other guides, but in most of the cases, as you can see here, we get very high efficiency, approaching 100% efficiency of editing. So this tells us that the delivery of the Cas9 reagents to the cell is extremely efficient with AMAXA to enable us to get almost every cell edited in these experiments. So just to drive home this point that guides are the wild card in these experiments, that we see variable activity between guides. This is an example of a project that we initially ran with a guide that failed. So we find the SG1 guide, which has a good off-target score, gave us very little editing. I think there's just one clone out of the whole 160 that showed NAGJ. All the rest of these were wild type. So we saw absolutely no editing with this guide. And so we selected a second guide, SG2, and we find with that guide we started to see good editing, good numbers of of heterozygous and homozygous clones. And the difference in the guides is shown below, which is they just offset a little bit, and the sequences are similar, fairly similar. They're fairly similar in GC-rich content. They're similar in their off-target score, but this just shows you how dependent these experiments are on the guide sequence itself. So we've now gone on to start to revert these mutations back to wild type, and so this shows you the reversion of autosomal dominant models that we've created, which are SNV heterozygous with SNV, and we're going to revert those to wild type, as I explained earlier, and we're getting good efficiency reversion of the SNV over wild type mutations using these optimized conditions. You can see the number of wild type clones that we're getting for most of these projects is very high. And it's also interesting that the efficiency of reversion does not necessarily correlate with the efficiency of the original editing. And I think that, again, emphasizes the point that the guide sequence itself, even a single base pair difference between guides, can affect the efficiency of these editing experiments. But nevertheless, we've been very successful in reverting these mutations back to wild type using a guide that recognizes the mutant copy. Okay, so the high efficiency of editing does create a problem for us, and that is that if we don't recover heterozygous clones from these experiments, how do we deal with this? And so what we needed was a method to help control the zygosity of these experiments. And we tried a number of different strategies, but the one that ended up working the best was just to simply add dead Cas9 to these nucleofections. The idea being that dead Cas9 with the guide will recognize the site, but it won't cleave that site. And so essentially it's protecting one of the two copies from cleavage and modification. So just by adding dCas9, we might be able to shift the outcome away from exclusively homozygous clones to start seeing heterozygous clones. And so this looked promising in our BFP assay. So you can see that in the BFP assay, with Cas9 alone, we get very high efficiency of HDR of the BFP to GFP. But when we add dCas9, we can shift the outcome back to wild type. So we get mostly unedited clones with the addition of dCas9. So the amount of dCas9 that you add is critical. And so we show this in this pilot experiment where initially we didn't get any heterozygous clones for this mutation. They were all homozygous. But by adding increasing amounts of dCas9, you can see a dose response where we start to see more and more heterozygous and more wild type clones after the addition. And so this turns out to be a very useful way to recover heterozygous SNV over wild type clones in those cases where we get very high RNP activity and exclusively homozygous clones. So we've done this over a number of different projects and trying to titrate the amount that we think will be most useful. So you can see that in all cases, we can shift the outcome of these experiments with the addition of dCas9. 1 to 4 is too much. You get no heterozygous or homozygous clones under these conditions. But if you scale back to 1 to 2 and or lower, you start to see more and more heterozygous clones. So from these results, we actually think that a 1 to 1.5 ratio of dCas9 to Cas9 is probably optimal. And so for the experiments that we conduct now, we basically run them under two different conditions with Cas9 only and with Cas9 plus dCas9, usually at a 1 to 1.5 ratio. And you can see that using from those two nucleofections on the far right, you can see that we get plenty of homozygous clones and heterozygous clones under these two conditions. Okay, so one of the really important QC assays that we run on these is to detect on-target effects. So a couple of years ago, Dominique Paquet's lab published a really important paper showing that in the case of editing, you can lose one of the two copies of the target, either by deletion or other rearrangements, or by this loss of heterozygosity, where the mutation breakpoint down to the distal end of the chromosome gets converted, and you see loss of heterozygosity. So these are events that we do not want. So in cases where you think you have a homozygous clone, you might actually have what is known as a hemizygous situation, where you have one copy of the gene intact, the other copy is lost. And so we wanted to be sure that when we are looking at a homozygous modification, and that could either be the homozygous SNV over SNV clones, or the revertants, which is a wild type over wild type clone, that we have both copies of the gene present. And so we developed LR-PCR sequencing, where we amplify the region around the target, which contains heterozygous SNPs for the cell line that we work with. This is called 2 heterozygous SNPs. And then we sequence those SNPs to make sure that they are still heterozygous in those positions. So this gives us a very rapid assay to determine whether we have on-target effects or not, simply by just Sanger sequencing. So this shows you the results for a set of homozygous clones for this one mutation in LRRK2. So this has got a homozygous mutation, G to A change, but we don't know if that has 2 copies or 1 copy, intact copy of the target. And by genotyping SNPs on either side of this that are amplified in this amplicon, we can determine that there is indeed loss of heterozygosity in some of these clones. So if you lose heterozygosity of the SNP, that is an indication of an on-target effect where the second copy is either deleted, or there's an insertion or a translocation, or there's been this loss of copy neutral loss of heterozygosity. And if you can see from the pattern of loss of the LOH, it can affect either side or just one side of the target region. The direction of the change can be in either direction. But it's very important that we screen these clones out by this assay. And so when we look at lots and lots of homozygous clones from our experiments, we actually see 7% of the clones carry on-target events, which is significant, but it's also lower than what was originally reported by Weisheit et al. So we're fairly satisfied that at least the optimal conditions that we're using is not creating a problem of on-target effects at the target region. We get a reasonably low level of these on-target events. Okay, so we've gone on and looked at these post-edited clones, not only by LRPCR, but by array and by G-banding. And the good news is that we see very little effect on genome stability in post-edited clones. So 98% of the clones that we've assayed show a normal array result, which means that we're not seeing a significant copy number variation in these clones or chromosomal abnormalities. We also run G-band karyotyping, and so far all of the clones that we have looked at, including revert clones, have maintained a normal karyotype. So that's very, very good news. And finally, as an indication of genome stability, we also ran an assay which looks at TP53 activity in post-edited clones using a reporter assay that we developed, which is shown on the right, which has 13 TP53 binding sites upstream of the minimal promoter drive and cherry. So we can transfect cells and treat them with doxorubicin and look at M-cherry activity as a measure of TP53 pathway. And you can see in all of the clones that we analyzed, which include the heterozygous, the homozygous, and the revertants, they all maintain robust TP53 activity compared, which is shown in red, compared to a control cell line shown in blue, which is mutated for TP53. So all of these results together show us that our optimal conditions for editing clones is having very little effect or minimal effect on the genome. So to summarize, what I've talked about is that we have really good methods now to generate precise single nucleotide changes in the genome, which is scalable to high throughput. And this has all been made possible by optimizing conditions, which I described, which include cold shock, HDR enhancer, and these N-modified single-strand oligonucleotides. But we've also found that these conditions are useful for other donors, like plasmid donors. And so I don't have time to talk about it today, but we're using these conditions as well to generate structural variants, specifically deletions of genes, and the introduction of protein tags using plasmid donors. And we're getting very high efficiency results from these experiments as well. So I'll stop there and just acknowledge people who have been working on this project with me. So we work in close collaboration with the Cellular Engineering Core at JAX in Farmington, Connecticut, with Justin McDonough, who runs the core service, and his staff. We've also hired a number of research assistants to help us with the picking and archiving and the genotyping of all of these clones. I also have a really very good research scientist in the lab, Gang Ning, who's helping this project and others. And we have had a lot of interaction with IDT over their HDR enhancer and modified oligonucleotides, and Synthego, which provide us with the guides that we use for these experiments. So I'm happy to answer any questions.
Thanks, Bill. That was an excellent presentation. We have a few questions from the audience. If anyone else has a question, please feel free to post it into the question box, and which appears at the bottom of your screen. So the first question is, what other HDR donors have you tried?
Yeah, I didn't have time to talk about this, but we've tried all sorts of donors. We've tried single-stranded oligonucleotides. We've short double-stranded donors. We've tried single-stranded DNA that we make ourselves. We've tried megamers you can buy commercially, which are single-stranded templates. We've tried circular single-stranded DNA templates and plasmid DNA templates. So for small changes in the genome, I think single-stranded oligos work beautifully, especially these N-modified ones. For larger changes, you require larger donors, and we find actually the best donor is still plasmid DNA. These other N-block double-stranded DNAs from IDT or the megamer, they work, but at much lower efficiency. So actually, plasmid donors are very, very efficient in the system, to the point where we can get high biallelic knock-in of, let's say, fluorescent tag or halo tag. So in our mind, if you want to make small changes that fit in an oligonucleotide, that's the best choice. And for larger changes, we think plasmids are the best choice.
Okay, great, thanks. And we have a question from Anthony Adamson, who is one of the keynote speakers. He asked, do you routinely use asymmetric SSODN donors?
Yeah, we do. We compared asymmetric donors with symmetric donors, and we varied the length of the donor and so on. And we find that the asymmetric donors are slightly better, maybe 5% better than symmetric donors, with 100 nucleotide donors. I don't think it makes a big difference, and the strand that you use doesn't make a big difference. So we just started making these asymmetric and stuck with it because they just give you a slightly higher, but not tremendously higher efficiency.
Okay, thanks, Bill. Next question, what if the gRNA does not overlap the mutation?
Yeah, this is the difficulty. So if you try to introduce a mutation on oligo using a guide that does not overlap the mutation, what happens is all of the clones are damaged by NHEJ. So we know that when you transfect Cas9 protein, although it only lasts for about 24 hours before it's degraded, within the cell, it's going through many cycles of cutting, repair, cutting. So if you don't block the cutting of the guide, it just continues to cut, get repaired, repaired until you've changed the nucleotide sequence so it can no longer cut. So with a non-overlapping guide and an oligonucleotide that contains your mutation, all you get are clones that contain the mutation but have also been damaged by NHEJ. So you don't get what you want from those experiments. So the way that most people deal with it is they would introduce the mutation outside of the guide and then the mutation in the guide to suppress this recutting that occurs. And that's fine, but for this project, we really wanted to avoid silent mutations because we're not really sure what their impact might be on the gene. So we did try dCas9 in these situations, and we have some limited data that suggests that adding dCas9 is also useful in preventing recutting once you've incorporated the oligonucleotide by HDR. So in fact, you can recover mutations with non-overlapping guides in the presence of dCas9. The issue there is that dCas9 also just reduces overall editing efficiency, so it becomes harder to get homozygous clones. But I think, with a little optimization, maybe trying different doses of dCas9, it is possible to introduce mutations outside of the guide sequence.
Okay, thanks, Bill. So the next question, in the first part of your talk, you spoke about how traditional cell lines are very abnormal, and it might be a good time to review what we've learned about the cell biology of iPS cells. Do you think iPS cells should or will replace the use of these traditional cell lines? And if so, how can the scientific community help to move towards this goal, and what are the challenges there?
Yeah, I hope they do move away from these highly abnormal immortalized cell lines. I feel passionate about that, and I've wondered about that for many years. I've tried to get cell biologists interested in stem cells in the past. Mouse stem cells also have the same properties as iPS cells, but I think the trouble is that you have to be a little bit more careful when you culture stem cells to avoid them differentiating and dying. So they're a little more finicky to work with than HEK293 cells, for sure. Also, the media that you use is more expensive. It contains expensive cytokines and so on. So I think that's also a barrier to using iPS cells, is that it just costs more. So unfortunately, I think people will still stick to these really highly abnormal cells. But I would really try to encourage people who want to understand normal biology to switch to a stem cell.
Right. Okay, just a couple more questions. You mentioned screening guides in vitro. How do you do that?
Yeah, so it's a useful step because you first test the primers that you're going to use for genotyping to amplify the region. So that's useful because not all the primers work well. So you amplify the region that you're targeting, and then you purify that on beads, and then you add in, into these assays maybe 100 nanograms of the fragment, and then you recombinant Cas9 and your guide, and the recombinant Cas9 RNP acts like a restriction enzyme and will cleave at the site that you've targeted to, so that you get cleavage products, and you just run those on a gel. So it's a very simple, quick assay, and I think it's worthwhile.
Great, thanks. Okay, so final question, Bill. You highlighted that guides are the biggest unknown, and that they have very similar features that can have dramatic differences in editing. Is there any other feature that you've noted that seems to correlate with better efficiency that is not commonly looked at, or is it just a case of testing different guides?
Yeah, so there have been some attempts at trying to identify the highly active guides computationally. So they've taken data sets where they have some data, and they've kind of derived some sort of computational score for guides that they think might be good, and those guides that they think might not be good. But we've looked at that score for some of these computational predictions, and while it's true that highly active guides do come up with high scores, so high scoring guides do work well. We also find that a lot of low scoring guides work equally well. So actually, you're throwing out guides if you use these computational prediction programs for guide activity. You're throwing out guides that are going to work very well. So I don't think we've yet cracked this issue of what makes a guide a highly active guide and what makes a guide a poor guide. I don't think we know the answer to that.
Great, thanks, Bill. So that brings us to the end of this keynote presentation.